welcome back to my channel. We are doing all the important chapters from ICSE 10 biology. Today let us understand all the experiments from absorption by roots chapter. I think most of you are finding experiments a little confusing, but it is not. It is very simple. Let's learn it under 5 minutes today. Okay, so the first experiment, before you go into any experiment, go through what is the aim for it. So what is the aim for it here? To show that roots absorb water. We all know that roots absorb water, but let's prove it scientifically today. So how do we do it? First, take a test tube, fill it with water, take another test tube, fill it with water as well. In the first one and the second one, if you notice, there is something called oil added here on top of the water in both cases, correct? Why is it done? Is your usual ICSE board question. It is done to prevent evaporation of water. We are trying to prove that the plant is absorbing water, but instead of the plant absorbing, what if water is lost through evaporation? We need to stop that. We need to eliminate that factor. Correct? So that is why we add oil onto the water. Alright, in both cases we added that. In the first case scenario, you're not adding anything extra. In the second case, you are adding a clean plant. When I say clean plant, you take a plant out of the soil slowly, wash the roots out of all the soil so that we can observe it very well. Alright, after 2 to 3 days or 1 to 2 days, what happens? Simple logic. What plant would have absorbed water, the water level will reduce. That's it. Hence proved. Okay. Experiment number 2. What does it say? It says that we have to show that water is conducted upward through xylem. Till now we know that water goes up. We learn the different ways in which water is going up, different factors and everything. How to prove that water goes up, especially through this group of tissues called as xylem. They are complex tissues. Remember complex tissues and simple tissues? Complex tissues are xylem and phloem. Xylem helps in conduction of water we have learned. Let's prove it today. Okay. So how are we proving it? First take a beaker, add some water into it, add a clean plant into it. Clean, what do I mean by clean again? Clean, the root should be cleaned without any soil. And if you want to observe something in a little colorful way, water is useless, isn't it? We need to have some color in it. How do you add color? You can actually add some ink into it and still hence prove it. But in the textbook, they have given eosin as the dye. EOSIN. It's slightly smudged here. Eosin is a pink colored dye. You add that to water and then keep the plant. So beaker, water, dye and then plant you're adding. Leave it for some time. After some time, take out the plant. Take a cross section of it, that is cut off the plant over here. Make one more cut so that you get a tiny thin cross section like this. Okay, this part you've taken out like this. What do you see? You see that every other part of the cross section that you are taking will be colorless for the except for the place where the colored water ran through. Right? Where did the colored water run through? It ran through a set of tissues called as xylem. We can easily observe that xylem ring is there in this experiment. Hence, we can say that the colored water that we made passed through this tissue and hence we can say that water is conducted upward by xylem. One more experiment is there. Aim remains same. That is conduction of water through xylem. We are again proving in this experiment we are using a different method though. Okay. What are we doing? We're taking two plant setups. What is the setup including? Beaker, plant, a stem connected to a stand. In both cases do the same. In first one, you cut out from the stem surrounding area. The central part you don't cut. Instead, you are etching out the side part of the stem. You keep on doing it till you get only the center part over here rest of the stem remains same. Okay, so the stem is there, only one selected area, you're carving out the outside part, go on carving till only the core is left. All right. Why are we doing that? It's because xylem is somewhat towards the core. When I say somewhat, what do I mean by somewhat? For that, go back to this diagram and see how is it. Where is xylem here? We have outermost layer called epidermis, then a group of cells called cortex, then endodermis comes, then pericycle comes, then comes phloem. After that, xylem. Xylem is, however, not in the core. You can see that the core of this is called a spith. A group of cells called a spith. Okay, here is we are not bothered about this. We are bothered about this xylem. So we are not cutting very thin. How are we cutting? We are carving out the stem in such a way that and quite an amount of stem is still there in the center. Okay, so that the xylem remains. That's what is done to the first setup. Second setup, what do? You do ulta. That is, in the core of the stem, the complete central part you are carving out. You can see here. All right. Now, leave both of them and check what happens to them. After some time, what happens is, in case of xylem, the one that you allowed xylem to stay, first setup, water is conducting upward, all the leaves are proper and turgid. What does that mean? They are plump, nice leaves. But in the second case, you can see this leaves have drooped out. They are kind of like this. Why is that? Because water is not going up. It's not getting any water. Okay. So that hence proved again. What did we prove? We proved that xylem is the conductive tissue for the plant. All right, one more extra information that you should know given in the textbook is that whenever you are doing this, that is carving out the outside part except for the central part, that is keeping the central part intact and carving out the outside part, you call that as girdling, Girdling. All right. Again, next experiment is to show that food from the leaves, you know that leaves are preparing the food, that is glucose, and it is transported throughout the plant. So that is what we are trying to prove through what tissue it's happening. Okay. So the aim is to show that food from the leaves is conducted downward through the phloem in the stem. How do we do that? We do the same experiment, that is, in the stem, we leave the central part for in this area, carve out the rest. And after some time, what happens? They have shown start and end. Okay, in the starting, the plant seems fine, the stem seems fine, but after some time, you will find that the upper side of the cut edge, right, this part starts swelling up. Why is it swelling up? Because whatever the leaf has made, it is transporting it downwards through phloem. But when it reaches over this area, no more phloem is there to conduct it down. Okay, so it gets stuck over here, resulting in a swollen stem. Alright, I hope it is clear. If you have any doubts, please connect with me. I'll be doing all the experiments from the upcoming chapters also. I know you're struggling with experiments. Let me know which experiment do you want me to do next. Thank you.